Very good morning, students. We are going to discuss that how the food is being digested in our intestine. We eat carbohydrates, proteins, and fats in our food, and it is being digested. It starts from the buccal cavity and adds into the small intestine. Now, how it is being digested? So let's start. So the food digested in the starts from the buccal cavity, that is mouth, with the help of the enzyme ptyalin, which converts the starch into the maltose. And when it reaches into the stomach with the help of hydrochloric acid, it hydrolyzes an amount of sucrose. The duodenum converts the starch and dextrin into the form of maltose sugar, whereas in small intestine, with the help of their juices, that is succus and taricus, sucrose, maltose, lactose, and finally they break down into the monosaccharides in the form of glucose and or the galactose, and dextrin is also converted to the form of glucose molecules. So yes, now we are coming to the absorption of the glucose molecules. As the monosaccharides, they are formed either in the gut or in the lumen. In the brush border of the epithelial cells are absorbed from the jejunum and upper part of the ileum, that is short our small intestine. And the process of absorption is either through the simple diffusion or it is an active transport. That is, there is no use of energy whereas it needs energy. Simple diffusion occurs when the concentration of sugar in the guts exceeds that is of blood. Whereas active transport, this causes absorption of sugars against their concentration gradients, hence it requires energy in the form of ATP molecules. So this absorption it requires, it is occurs in two steps. The step first, and both these steps they require sodium potassium pump channel. They use the sodium potassium channel. The glucose combines with the carrier, a mobile component of cell membrane, to form glucose carrier complex. You just can see this here. This is the glucose carrier complex, which moves the glucose across the lipid barrier or the cell membrane and releases the glucose molecules inside the cell. And the sodium ions have the same carrier. Similarly, the sodium ions, they also have the same carrier and they enters inside the cell with the glucose. And this method is known as symport mechanism because both are moving in the same direction, and this is known as symport, that is SGLT1, right? And this step requires no energy. Now we are coming to the step number 2, that is GLUT2. To concentrate the glucose within the cell, the carrier, that is sodium glucose transporter, is coupled to form to a source of energy. Once this SGLT, it couples to a source of energy which provides indirectly by active transport of the sodiums out of the cell into the intracellular spaces. Just come here. The sodium, it couples with the source of energy, that is, and it in the presence of ATPase enzyme, it enters from cell to blood capillaries. The glucose is transported by glucose transporter 2, that is GLUT2, that is second step into the interstitial spaces and finally then into the blood capillaries. The maximum rate of glucose absorption from the intestine is 120 gram per hour. The rate of absorption of monosaccharides is variable. It is higher for the glucose and less for the maltose, sucrose or other sugars. So this is the ultimate fate of the glucose molecules, that how they are being absorbed through the passive as well as through the active transport inside our blood capillaries. This is the overall summary of the carbohydrate digestion and absorption. Here is the sodium and potassium pump channel which requires some amount of energy. The ATP molecules in presence of ATPase converts into the ADP molecules. And this is, here you can see, this is the antiport mechanism. The sodium is going out inside the cell blood capillaries, and whereas the potassium ions from the blood coming out inside the interstitial spaces of the cell. That's why it requires some amount of energy. Yes, now we are coming to the next digestion of the fat molecules. As you know, there is no digestion occurs inside the buccal cavity of the fat molecules. There is no lipase, and very lipid starts the digestion from the stomach, as there is a very weak lipase, and it converts the few amount of fats into the fatty acids and glycerols. The ultimate digestion of the fat starts from the small intestine with the emulsified uh, salts which comes from the bile juice, that is sodium chlorate and sodium taurochlorate. And these emulsified fats further being converted into the fatty acids and triglycerides with the help of pancreatic lipase. And the disaccharides are again being break down into the fatty acids and monoglycerides. And monoglycerides may be converted into the free fatty acids and glycerols. Glycerol is a water soluble compound, so easily being absorbed by the intestine. But the absorption of the fatty acids is somewhat complex. Now let's see how they are being absorbed. Absorption of the fats. The pancreatic electrolytes, that is monoglycerides, fatty acids and bile salts, they interact spontaneously to the polymolecular aggregates called micelles. Here you can see the bean salts are coming from the pancreas as well as from the bile cells, and they combine with the fat molecules and they form this complex emulsified, that is micelles. These are the micelles, and they are water soluble complex and 3 to 10 nanometer in diameter with variable lipid concentration. They can dissolve hydrophobic compounds in their interior, and such constituents as cholesterols are therefore made water soluble. Thus, micellar formation for further solubilize the lipids and provides a mechanism for their transport into the interstitial cells. So these the micelles, they enter through the brush border inside the interstitial cells. So they are actively reabsorbed from high concentration to lower concentration. The fat contents of the micelles consist of monoglycerides and fatty acids. Once inside the intestinal cell, they enter. Now here there are some complex uh, reactions occurs. These micelles inside the Golgi body combines with the protein molecules and they form further the droplets known as chylomicrons. Here you can see the fatty globules, they combine with proteins to form chylomicrons inside the Golgi apparatus. And these chylomicrons are being excluded from the epithelial cells and enter into the lacteal vessels, cells enter, that is the lymph capillaries. So this is the ultimate absorption of the fat molecules. But again, if these fatty acids or the monoglycerides are greater than 4 carbon atoms, are re-esterified to triglycerides into the mucosal walls, and these are known as chylomicrons. The esterification of the fatty acids occurs if they are more than 14 carbon atoms. But if they are less than 14 carbon atoms, these micelles, they directly enters into the blood capillaries. They are known as non-esterified fatty acids, and they forms the triglycerides and cholesterol in our blood capillaries. So this is all about the fat absorption, that how the fat molecules are being absorbed. On a moderate fat intake, only 5 to 6% of total fat per day is excreted in stools. However, the process involved in fat absorption is not mature at birth. Therefore, infants' fecal fat contains is 10 to 15% of ingested fat. The fat is being absorbed only in adults. Now we are coming to the blood fats. The carbon atoms which are less than 14 carbon, they directly enters into our blood. The plasmolipid, that is triglycerides, free fatty acids, phospholipids and steroids are bound to plasma protein to form lipoproteins. They combine with the plasma proteins and they form lipoprotein complexes, which are very in density according to their lipid constituents, has been divided into the LDL and HDL and VDL. That is very low density, high density lipids and very low density lipids. The very low density lipoproteins, that is VLDL, they have a very low density, and the LDL, the low density lipoproteins, The serum triglycerides concentration bound to lipoproteins, that is 30 to 150 milligram percent, whereas serum cholesterol, they are approximately 150 to 240 milligram percent in a normal person, and serum phospholipids lecithin and cephalins 150 to 300 milligram percent. They are bound to alpha lipoproteins and form LSDL, high density lipids. Serum FFA, that is, they are approximately 10 to 30 milligram percent is found in the normal human beings. So this is all about that how the fat molecules are being absorbed. So LDL are very harmful for the human body as they are the low density lipids, and they very easily settle down in our blood capillaries and they can choke and cause the heart attacks. So now we are moving to the absorption and digestion of the protein molecules. The digestion of the proteins starts from the stomach with the help of hydrochloric acids, converts the proenzyme pepsinogen into the pepsin, and similarly the renin, that is the curdling of the milk, and the enzymes here this chart is showing that the glycerin is an hormone which controls the secretion of overall stomach, whereas secretin controls the overall secretion of the intestine, cholecystokinin, pancreazyme, that is CCK and PG complex. It controls the secretion of accessory digestive glands. So that is the overall the summary of the digestion of the carbohydrate, proteins as well as of fats. Now we are coming to the absorption of protein molecules, that how these amino acids are being absorbed. The D amino acids are absorbed by passive diffusion, simple by the passive diffusions, if they are D amino acids. While L amino acids are actively transported, they need some amount of energy. They are being need in the form of ATP molecules. Absorption of both D and L isomers is coupled to a sodium transport channel. Here again the sodium transport channel is required, and once the sodium and potassium channel is required, then definitely the energy involved in this process. The transportation of amino acids accumulate into the mucosal cells, here into the mucosal cells, and they enter through this carrier proteins, that is sodium and potassium channel. Here is the peptide bond, and they enter, that mechanism involves both symport and antiport mechanism. Some of the ingested proteins enters the colon and are finally digested by the bacterial action. The protein absorption decreases with the age, whereas in infants it's very high, but as well as the age in the old age, because the absorption of the protein is very low. The absorption of water molecules in the it presents each day with approximately 2000 ml of ingested fluid and 7000 ml of secretion from the mucosa of gastrointestinal tract associated with our glands. So 90 to 95% of this fluid is reabsorbed with a daily fluid loss of only, that is 200 ml in the stools, and this amount of the absorption of water occurs inside the ileum, that is the small intestine and colon, uh, against the osmotic pressure. So that's all about the absorption and digestion of carbohydrates, proteins and fats. And here again a summary of all the digested food and absorption. Here is the process of digestion, and this hope you will understand well digestion and absorption of carbohydrates, proteins and fats. Thank you.